Namaste students, I am Jibin Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic genetics of immunoglobulins. Uh, in the previous video, we have discussed about immunoglobulins, their structure, their functions, the types, monoclonal antibodies, hybridoma technology, and their productions. But if we talk about the genetics of immunoglobulins, then we not only go to the immunology, but also we go for the molecular biology also. It covers the molecular biology subject also. So genetics means the genes which are involved in the production of a particular kind of an antibody. Genes, we know that a particular kind of genes is involved for and it is responsible for the particular kind of protein. Okay. There are several genes which are present in our body, and similar type of genes are also there which are responsible for the antibodies and antibodies productions. So this is not enough for the production of immunoglobulins, but their expression is also very important. And how the immunoglobulins will be expressed, their expression will depend on the immunoglobulin genetics. There are antibody diversity. What is the need of the immunoglobulin genetics? The need of immunoglobulin genetics is because antibodies are diverse. Antibodies are diverse, different antibodies are required. We know that the antibodies must vary and they need to be vary. What is the reason of their variation? There are millions of antigens are present. Millions of uh, microorganisms are present, and these microorganisms have millions of structures, millions of epitopes. And if we talk about these epitopes, then their identification is very difficult. And who will identify them? Only single type of antibody can identify them. No, they cannot identify by a single type of antibodies. And which region is responsible for their identification? The variable region of the antibody is responsible for the identification of the antigens. If we talk about the millions of antigens, then we must also talk about the millions of the antibodies also. But is this possible? Because in a single human body, we can produce millions of antibodies. Is this possible? It is not possible in a constant form. But when an antigen enters into the body, it is possible because the antigen triggers the immunogenicity. Antibodies are triggered for production of the antibodies. And this is due to the some genes organizations which are present on the antibodies. So what is the gene organization? One of the most remarkable features of the immune system is the ability to respond foreign antigens, and the genetic basis of the such variation is coupled with the consistency in a single protein molecule which lies in the organization of the IgG genes. And and IgG is a tetramer of the two light chain and two heavy chain. There are two light chain and the two chains. So if we talk about the light chain, we also talk about the heavy chain genetics, two genetics, heavy chain genetics and the light chain genetics. Each chain has a N-terminal variable region, which is V region, and the C region, constant region, and the variable region. V region, V domains, and C domains are separately coded by the V gene segments and C gene segments. V constant region have a segment gene which is known as C gene, and the V gene variable region is encoded by the V gene. But V and C gene segment is not expressed as an independent unit. To construct a unit that cannot be expressed in the form of the light and heavy chain, a V gene segment must be joined to a C region. So in the antibody, if these are the V region and this is the C region, so V region and C region must be bound to each other because they are part of the antibody. So genes of the V regions are different and the genes of the C regions are also different. Each kappa and light chain. If we talk about the light chain genetics, each kappa and light chain polypeptide consists of two major domains, a variable region and the constant regions. So in the as we were talking about the heavy chain, there were a variable region and the constant region. In the light chain also there are a variable region and the constant region, VL and CL, and which are coded by a particular gene that is the V segment and the C segment. And the V segment and the C segment are joined by a middle segment which is known as the J segment. J. And this J segments also help in providing diversity to the immunoglobulin. It also helps. What is the function? Another function of J segments? It is J means joining segment. Now talk about the genetics. First, we will talk about the genetics of the light chain. If we talk about the genetics of the light chain, the kappa locus is found on the chromosome number 2 in the human. Chromosome number 2. And there are approximately 40 different V kappa chain. V kappa chains, each of which can code for the N-terminal amino acid sequence of the kappa variable regions. So there are 40 genes. If there are 40 genes, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 40. Is it possible to express all these 40 genes for a particular antibody? No, this is not possible. Only one gene will be required for encoding a for the production of a particular antibody. Only one gene will be required, not all the 40 genes. Okay. Similarly, if we talk about the J region, there are 5 J genes, J kappa genes. These are another one more important thing is that for the kappa chain and for the lambda chain, the genetic diversity is different. we need to understand the genetics of the kappa chain and the lambda chain separately. So we were talking about the J chain. So J chain also has 5 genes, okay, which are JK1 to JK5, JK1, JK2, JK3, JK4, JK5, and then next constant. Okay, so in the light chain, first V1, V2, V3, V4, V5, V6, V7, V8, V9. So this is the sequence of the VL genes, and then the chain of the VJ chain sequence, JK1, JK2, JK3, JK4, JK5, JK5, and then it will continue from the constant region. I must explain it with an example here. So this is the gene sequence of the kappa chain of light chain. Light chain, kappa chain. So this is the double stranded DNA, light chain, and V genes are there. 40 V genes are there, and 5 joining genes are there, and then it is constant region genes. V1, V2, V3, V4, V5, V6, V7, and then a gap is there, and G region genes. J1, J2, J3, J4, J5 genes are there. Okay. Same thing is in the lambda chain. In the lambda chain, the lambda locus is found in the chromosome number 22 in the humans. And there are 4 constant region genes. Okay. And approximately 30 variable genes are there. Only 30 variable genes are there. And J regions genes are also there. J1, J2, J3, J4, and then it will continue from the concentration 1, concentration 2, concentration 3, concentration 4 genes. So this is the sequence of a particular immunoglobulin in the light chain. So I hope that students have already been understand what is the gene organization of the um, light chain. Now we must need to talk about the gene sequence of the heavy chains. If we also talk about the heavy chain segment, there are very much more gene sequences as compared to the light chains. So there are 3 regions: V region and J region in the light chain. But in the heavy chain, there are 3 regions: V region, D region, and the J region. In the V region is for the variability, here D region is for the diversity, and J region is for the joining. J for joining. The D region provides diversity of the immunoglobulins. The H chain locus is present on the chromosome number 14, and the number of V genes are there 50, and the number of D genes are 20, and the number of J genes are 6. are followed by the concentration genes, and then the types of uh, heavy chains, mu chain, lambda chain, gamma 3 chain, gamma 1 chain, alpha, gamma 2, gamma 4, epsilon, alpha 2. So these are the chains which will decide the type of antibody to be prepared. So this is all about the normal genetics of the immunoglobulins. So we have discussed here, we have learned about the gene organization of immunoglobulins. What are the genes which are present in the immunoglobulins in the light chain and the heavy chains? And in the next lecture, we will discuss about the rearrangement, how the genes are responsible for the repression of the particular antibodies, how antibodies will be prepared from these genes. So students can be able to understand the genetic organization, what are the genes which are involved in the immunoglobulin structure, and they can also understand the diversity of immunoglobulins. And which kind of questions can be asked here? The questions which can arise here that explain the genetics of immunoglobulins and the genes which are involved in the light chain and the heavy chains of immunoglobulins. So my lecture is over here. Thank you very much.